Okay, great. Thank you, everyone. Thanks for bearing with us as we start just a few minutes late. I think everyone is used to working with the different interfaces. Okay, so welcome to the Kids First Fall Public Webinar. Thank you for joining today. I would like to remind everyone that it is being recorded because I hit the record button. Now that we are in October, we've officially just completed 6 years of the Gabriella Miller Kids First Pediatric Research Program, also known as Kids First. Today, after a brief introduction from us, you will hear about progress on Dr. Sharon Pong's Cancer Susceptibility Project, and Dr. Pong will address questions right after her talk. Then we will get updates from the Data Resource Center before hearing about the recently launched Common Fund Data Ecosystem and how they are working with the Kids First DRC. And we will also share some program updates before addressing common questions and opening the floor to additional questions from you all. My name is Valerie Cotton from the Eunice Kennedy Shriver National Institute of Child Health and Human Development, and I am the program manager for Kids First. And I'm here today representing the larger Kids First Working Group, which is overseen by the NIH Common Fund, chaired by leaders from NICHD, NHGRI, NHLBI, and MCI, and includes representation from 12 other institutes and centers. For those of you who may be new to Kids First, the program was initiated in response to the 2014 Gabriella Miller Kids First Research Act, which transferred $126 million from a presidential nominating convention fund to a pediatric research initiative fund and authorized the appropriation of $12.6 million per year for 10 years to the NIH Common Fund, with the first appropriation in 2015. Gabriella Miller, the bill and the program's namesake, was a 10-year-old advocate for pediatric research who lost her battle with diffuse intrinsic pontine glioma, or DIPG, just 6 months before the bill was signed. The vision of the program is to alleviate suffering from childhood cancer and structural birth defects by fostering collaborative research and uncovering the etiology of these diseases, and by supporting data sharing within the pediatric research community, which we believe will accelerate pediatric research and ultimately lead to the development of improved diagnostics and therapeutics for patients and their families impacted by these conditions. Childhood cancers and structural birth defects have profound lifelong effects on children and their families, and several publications, including this one from Sharon Pong and Philip Lupo's group, have revealed associations between childhood cancers and birth defects, suggesting that there are shared genetic pathways underlying these conditions. But since individual childhood cancers and structural birth defects tend to be relatively rare, there was a need for coordinated efforts to build, combine, and study sufficiently powered data sets. To respond to this need, starting in 2015 and continuing through this year, we are whole genome sequencing childhood cancer and/or structural birth defect cohorts. And since 2017, we have been developing the Kids First data resource to facilitate and empower analyses of these data sets. And during the program update section at the end, I will tell you a bit about our plans for the remaining years of the program. To date, 40 childhood cancer and structural birth defects cohorts have been sequenced or are somewhere in the sequencing pipeline, and 14 of these data sets are accessible to the research community. The data are or will be shared through the Kids First Data Resource, which is a cloud-based platform designed to empower and accelerate collaborative pediatric research. There are multiple components of the resource, but the two most important to this community are the Data Resource Portal, which is where researchers and the public can view and visualize Kids First data and discover which data sets you want to learn more about or analyze based on your scientific questions. And the cloud-based workspaces in Incubatica, which is where researchers can pull together and co-analyze multiple disparate data sets. Of course, you'll hear more about this from the DRC's presentation today. More than 1,700 users from across the country and the world are accessing the portal, and more than 150 data access requests have been approved for external researchers to analyze whole genome sequence data from the 14 available data sets. And researchers are analyzing these data with support from a variety of NIH grants. Although Kids First does not fund analysis grants out of its appropriated budget, institutes and centers from across the NIH support this investigator-initiated work through a variety of mechanisms, including an R03 that is specifically designed for supporting analysis of Kids First data and associated pipeline or tool development. And we are happy to see that many of you have been successful in competing for these awards. And discoveries are emerging from these analyses, such as uncovering new genetic causes of childhood neuroblastoma, identifying genes or regulatory networks that are important to the development of the diaphragm or the heart, and identifying genetic variants that lead to craniofacial anomalies in different ethnic populations. You've heard from some of these investigators in the past, and we will continue to highlight these findings in future webinars as well. And without further ado, allow me to introduce our featured Exo-1 speakers for today. Dr. Sharon Pong's research has focused on understanding inherited susceptibility to childhood cancer, as well as other congenital anomalies. She's a leader in high-impact initiatives such as CSER, or the Clinical Sequencing Exploratory Research Project at Baylor, and ClinGen, and serves on NHGRI's Advisory Council. In the spring, you heard a bit about her from her student, Owen Hershey, and how he's used Cfde to develop and run workflows to analyze this Kids First data set, and I believe you'll hear a little bit about the progress of his structural variant analysis today. So with that, I'll help unmute you, Dr. Pong, and I will hand over the presentation.
Great. So you'll let me share. Well, thank you. That was actually a great intro. I didn't know you were going to do that, although I didn't realize how many years we were in already. I'm just going to share my screen to make this. Yes. Sorry, I'm. Okay. So. I did just want to comment, since you gave the history, that I had the honor of meeting Gabriella Miller's mother, who early meetings when Kids First was getting underway, and really just talked about the importance of this work. So great to hear the overall progress. So that should look correct. Does that look right? Yes, Valerie?
Great.
Okay, so I'm going to talk about identifying novel cancer susceptibility mutations from unselected, and I want to emphasize, that'll be sort of a theme I emphasize, childhood cancer patient and parent trios from the Basic 3 study. So I'm going to give a little bit of background about what did we know about these families based on our CSER project. That's where they came from. And then what did we hope to learn with whole genome sequencing? And then Owen's going to give an example of a very recent, totally unpublished finding that we're excited about pursuing. So just a disclosure that I'm on the Baylor Genetics Scientific Advisory Board for their laboratory. So really, there have been multiple different approaches to understanding the genetic basis of childhood cancer. And I'm going to briefly talk about what's been going on most recently with regard to our Basic 3 study, and I won't talk about the subsequent study. I think you're all aware, and is emblematic of Kids First, of most of the Kids First projects are sequencing large cohorts of specific pediatric cancer patients. So the neuroblastoma discovery that just got mentioned came from one of the Kids First neuroblastoma cohorts. And we won't talk about it today, but another area where we're really learning more about genetic childhood cancer is from precision oncology trials that are really designed for treatment trials, but are now beginning to do matched paired germline and tumor sequencing. So the NCI Children's Oncology Group Pediatric Match Trial does do a germline panel along with the tumor panel. And I think they are looking to now, on a research level, do whole genome sequencing from that cohort as well. So as was already mentioned, this project started as part of an NIH initiative called CSER, the Clinical Sequencing Exploratory Research Consortium. It's now still CSER, but now we're the Clinical Sequence Evidence Generating Research Consortium. So I just want to say that the original project was a very large collaborative effort across Baylor, sequencing, ethicists, geneticist, and Will Parsons, who is a pediatric oncologist, many of you know, who's been my partner throughout these studies. So I just want to spend a little bit of time on this study because it really just highlights who are the patients we're talking about. The Basic 3 trial, which started enrolling in 2012, enrolled sequentially newly diagnosed unselected patients. So they had to have a brain tumor or a non-CNS solid tumor. The family had to speak English or Spanish. Those were the only, and it had to be a malignancy. So if a patient had a lipoma or a benign neurofibroma, that was not included in the protocol. But otherwise, we took all comers, and I'll show you what that mean what that looked like in a couple of slides. We isolated frozen tumor and blood from both the patient and both parents. And we did CLIA-certified whole exome sequencing in a clinical laboratory that was a joint effort of the Human Genome Sequencing Center and the Baylor Clinical Diagnostic Lab at the time. And we generated a germline exome and a tumor exome report for each patient. If a mutation was found in the child, then it was validated in the parents, but we did no sequencing of the parents otherwise. The results went back to the electronic medical record. They were disclosed to the family by their oncologist and a study genetic counselor, and we followed them. And the goal was really, and could you do it? Remember, this was starting back in 2012. And could do it, what were the overall results of incorporating germline and tumor sequencing into pediatric care? So there are a couple of different, and just to say the methods were all published in our original publication. We're halfway through the study, but I'm going to give you final results, which sadly have not yet been published. So there are a couple of ways to think about diversity, and I think this is something we don't talk that much about at Kids First and probably worth looking at, which is what is the underlying diversity of the patients that we're enrolling in these cohorts. And so we were very proud of the fact that, as many of you know, Houston's a very diverse city. And this study, which was published, and then these numbers are actually updated once we completed the study, or close to completion of the study, was that there was really no difference in the decline rate. So the patients who entered the study represented our population. And you can see here the study overall, and the final study, I believe, was 48% Hispanic. And it was about 10% African American. And then there was the majority of the other patients were non-Hispanic whites. But it was a diverse patient population underlying their ancestry, underlying some of these findings. Another way to think about diversity, and this is particularly relevant to the study, that to people who want to use our Kids First data, is this is very much unlike most of their Kids First projects. We took unselected patients, so Will took the first about 150 patients and made these beautiful pie charts. I don't think he ever completed them for the whole 282 patients. But you can see we have a wide variety of malignancies. We have both the ones you would expect that are more common, neuroblastoma, Wilms tumor, but we had individually very rare diagnoses, and I'll comment on some discoveries we made out of that. And that's true of both the CNS and non-CNS patients. And then finally, although I won't talk a lot about this, was the diversity of what we returned. So every patient had a tumor report with every somatic mutation that was in a coding region. And then they had these 5 classes of germline results. The one that will be relevant to our discussion today are the cancer findings. So I want to just briefly go over the cancer findings and then what our plans were for Kids First. So of the known cancer genes, so these are molecular diagnoses. So these had to be known genes. What is it? About 10% of the patients carried a germline finding. And our surprise was, first of all, even though it's only 27 patients, it represented 19 different genes. So although P53 was more common, it was not the major outlier. We saw it 3 times, as did we see VHL. But we saw a wide variety of individual genes, many of which were known to be associated with the tumor, DICER1 and a pulmonary pleuroblastoma patient. But surprisingly, almost 40% of these findings were in genes that were not to be associated with the patient's tumor. And this has been quite interesting. We're not alone in this early. St. Jude published their large series a few months before ours, and they also reported BRCA1 and BRCA2. As I think many of you are aware, this has now been looked at in several additional cohorts where there were large enough cohorts to do sort of case control analyses, including our study that's not yet published, but soon to be on rhabdomyosarcoma. And it does seem that BRCA2 particularly may be overrepresented in pediatric cancers, and I think that's something interesting. Another thing to think about when you're analyzing Kids First data, which was very sad to me because I spent much of my early career studying rare recessive disorders like Rothman-Thompson, is that almost all studies that have been published so far, Bi-Allelic mutations in recessive disorders are extremely rare. So one of our 300 patients had a disorder that I'm going to talk about in a minute related to hepatocellular carcinoma. The St. Jude study, again, I think of their 1,100 patients, they had 2 or 3 at most. And I think it's important when you're looking at the data to contrast this with the fact that single pathogenic variants in recessive cancer genes are quite common. So we found about 0.5%. I think St. Jude found around similar numbers, maybe 7%. And the significance of those single pathogenic variants is really unclear. Again, there was a study recently suggesting that single RECQ-L4 variants in osteosarcoma may be significant. But again, when you're analyzing the Kids First data, you will find a large number of single pathogenic variants in recessive disorders. But the contribution of truly bi-allelic, a recessive diagnosis in pediatric cancer seems to be very, very low. Okay, so why do whole genome? And why do whole genome from such a heterogeneous data set? And I will say the original reviewers of my EXO-1 application had a similar number of questions. So we were delighted when we were selected despite that concern. And I just want to give 2 examples that came out of our exome data of why it's important to remember that even if we are sequencing heterogeneous data sets and we may only have a few patients with that type of tumor, that if you do find something, there are other data sets or other rare patients that you may go to confirm it. So these are actually 2 papers that came out of Basic 3, both from single patients in the study. So I mentioned already that we had a single patient with a bi-allelic. This patient was homozygous for a frame shift in TJP, tight junction protein 2, and had hepatocellular carcinoma. And through one of our collaborators at Texas Children's, Dr. Lopez Terrada, who studies pediatric brain tumors, and Dr. Feingold, they identified that they had a collaborator who actually also had a patient with hepatocellular carcinoma and had this otherwise extremely rare inherited disorder of liver function. And in fact, we were able to combine the data, look at the pattern of pathology in the livers and in the tumor, and publish this, and subsequently actually several other patients with TJP2 deficient cellular carcinoma have been reported. On the right is probably an even more remarkable study. We had a single patient with clear cell sarcoma of the kidney, actually had a stop loss predicted by the exome. But Angshan Roy, who was reading them out, was sort of surprised by that. He actually pulled the reads, and actually it became clear that in fact it was only a stop loss because there was an internal tandem duplication at the last exon, at the last codon. And he actually was able to pull tumors from the TCH pathology collection and actually showed, I think it was like 8 of 10 had internal tandem duplications of BCOR, and then was actually able to confirm that in the target data set. So again, an example of a single unusual result in one Basic 3 patient that then led to follow-up studies. So I think really that, in a way, was our justification for saying it's worth taking this data further. So to kind of quickly fill the goals of our, the goals of our Kids First project were to utilize the rich existing data set from Basic 3 to make further discoveries using whole genome sequencing of the proband and the parent trios. So just so people are aware, because this is available to you, the germline exomes of all the probands have been submitted to Dbcap. The tumor exomes from the majority of the probands, I think 70 or 80% of the probands had tumor exomes have been submitted to Dbcap or in Dbcap and being used by others. We actually do have RNA-seq for just a subset of the tumors. We DNA. We did, though, have DNA from the parents and the patients. And these blood samples were very high quality DNA. And that actually gave us great whole genome short read data. And as you'll hear, was the reason we applied to be part of the long read. And we actually also have lymphoblastoid cell lines. So every patient and every parent in basic 3, we endeavor to make LCLs. So we actually can grow cells, we can do functional assays, we can do splicing studies as needed. So how are we approaching the kids' first analysis? Obviously, we're doing trio sequencing. And we prioritized complete trios with adequate DNA, obviously. We did have some patients with more unusual histories, multiple malignancies, a few patients with birth defects. The study that I did with Philip Lupo, I think it'd be great to have Philip come and talk about that study. That was a population-based study and not based on basic 3. We did actually include a few solved cases. So, for example, a parent with a P53 mutation, because we actually wanted to look at how many did the child have. And we did that analysis. I have to say, I don't think there was anything remarkable that came out of it, but it was one of our original goals. So generally, our analysis has focused on both single nucleotide variants and structural variants. We've prioritized de novo variants in both situations. However, we have also analyzed known cancer susceptibility genes, looking for any missed variants that might have been missed from the exome. And more recently, we performed Splice AI, which is a relatively new artificial intelligence algorithm to try to predict potential missplicing events. So we've also done that with all of our kids' first data. So far, that hasn't yielded anything too interesting. So just briefly, so I can turn it over to Owen, talk about structural variants. We did de novo SNV calling. I will say this is where the kids' first team has been extremely helpful. We've already this platform on our genome human genome sequencing center data, and we've been using Platypus, which was a paper that was published in Nature Genetics a couple of years ago, which was specifically designed for trio analysis. And so we were able to work with the kids' first team to bring Platypus as a tool into Chematica. And so obviously our prioritization was pretty standard. We prioritized, in this case, de novo coding variants, although I will say we have gone back and looked at all known cancer susceptibility genes, see if we missed anything. We determined, obviously, the predicted functional effect of the variant. We ranked them based on things like CAD, and we also did sort of hand mining of the medical literature for them. And we got kind of the typical, got genome-wide, 60 under it for most of the trios. There's one trio that has more de novos, which we've tried to explore. We don't have a good explanation with, and not surprisingly, 0 to 4 de novo coding variants. So this data has been interesting. I would say the main de novo variant from all cohort that we've been looking at is heterozygous missense variant KMT5C. It's rare, it's in a highly conserved functional domain, has an extremely high CAT score and a high REVEL score. The patient has Appendamona, which, as you'll hear, Owen is interested in generally and has an interesting structural finding. And is an example of a variant in a chromatin remodeler. And chromatin remodelers play a huge role in pediatric brain tumors, so that's particularly interesting to us. So that's sort of where the SNV data stands. And then Owen, I think, is the next slide, is just going to talk briefly about his structural variant work. He'll repeat a little bit of what he said in the spring for those who missed his talk, but then he'll show you kind of an interesting new result. I can just advance the slides if you can unmute him.
Yeah, that would be great.
Okay, we can hear you.
Okay, great. Well, hello all. I'm Owen Hershey, one of Dr. Klein's graduate students at Baylor College of Medicine. And today I'll be talking about the structural variant analysis in the Kids First Basic 3. So a little background on structural variants. They're commonly reported in cancers, and some are known to play critical roles in said cancers, such as the Philadelphia chromosome translocation in leukemia. In the Plon lab, we've actually recently reported on germline structural variants in pediatric cancers, such as a balanced inversion of the APC gene in a pediatric hepatocellular carcinoma. And this was actually discovered through RNA sequence analysis. However, identifying these germline events in short read whole genome sequencing is challenging. Since 2010, over 40 programs have been created to identify these structural variants from short read whole genome sequencing. However, they were primarily built for identification in tumors, which requires the elimination of germline events in the results. And each of these individual programs have low sensitivity and a fairly high false positive rate. So that's where we believe we can kind of create a better pipeline to identify these germline events. Next slide. So for our pipeline, we actually want to use 5 different structural variant columns. So these are all available on the Kids First Cavatica platform, and they're mentioned below. And so what we do is we run these 5 different structural variant collars on each member of the trio and generate a list of variants identified per individual. Then we use various sliding filters and visualization methods to dwindle down which are false positive and which are true positive. And so what we found is using multiple structural variant collars has allowed us to increase sensitivity and specificity in variant identification. And this was tested on an independent childhood cancer cohort. Currently, this pipeline is built for the identification of de novo structural events, but we're now beginning to tune it for inherited variants. And so I want to talk a little bit about how we use this pipeline on Cavatica. So Cavatica has been very cool in that you have 2 real modes of operation to run tools. First is through application-based, and the next is through terminal-based. So for the application, we're allowed to run up to 80 parallel tasks at once, which in comparison to your local system, you may be able to run up to 8. So if you think about it, when we run 5 different structural variant collars on 165 different samples, that saves on quite a bit of physical hours when completing this. And these applications are tunable just in the same way you would tune them locally. And then once we generate those variants, we can analyze them in the interactive analysis cruncher feature of Cavatica. So this allows us to run complex bash scripts for filters and visualization tools and allow us to identify which are true positives. And it's very similar to what a user's terminal would be running these bash scripts. Next slide. Yeah, so just to talk a little bit about the filters we do use, we first filter out those structural variants that are found in poorly mapped regions of the genome or low complexity regions. And right now, we've just looked at de novo events. So those are only occurring in the child and not in the parents. Then we filter against a healthy population to determine for rarity. And then finally, just to determine these are true positive, we visualize the breakpoints in IGV or integrated genome viewer. And the outcome of this manual visualization process on our 54 program trios, we identify anywhere between 0 to 1 de novo structural variant per trio. This fits in line with published literature on what you would expect to find in general population. So we identified 10 deletions, 7 duplications, and 0 inversions in the 3 cohort. We're currently looking to identify these translocations, but that's still a work in progress. So a couple variants stick out, and so the first one I'm going to talk about is this germline de novo heterozygous 20 kb deletion of NF2, which is a known cancer predisposition gene in a meningioma patient. So what we're looking at here is the visualization of next generation sequencing data in IGV, which shows where reads map to on the genome. So we identified this deletion, which can be denoted by the drop-in coverage you see in the proband. This deletion occurs in exons 5 through 9 in the NF2 gene. And what's kind of interesting about this mutation is, unbeknownst to me, it was previously identified when this patient was first diagnosed. This was done through clinical sequencing of NF2, and they identified it as a potentially somatic mosaic event. And so we can kind of show that this is also potentially mosaic, as we only see it in 6 of the 40 reads in IGV. And this is important to us because it shows that our pipeline can find true positives as well as potentially mosaic events. Sorry, next slide. So the next one I want to talk about is a particularly interesting variant to me because of the germline de novo heterozygous 8kb duplication of the prostaglandin reductase 2 gene in an appendamoma patient. So what we see here is an increase in coverage in this highlighted region within the proband. And from the whole genome sequencing data, identify this as a head-to-tail tandem duplication. And one of the more interesting facts about this is it overlaps the promoter and first exon of all transcripts of the PTGR2 gene. So PTGR2 is actually a putative oncogene in brain tumors, and it's expressed. And the reason this is interesting as well is because this increase or this duplication may lead to an increase in transcription. Right, slide. So on initial analysis of this variant through ENCODE, we can see this hypothesis may be correct. So what we're looking at in ENCODE is whether, or they identify regions where there's open chromatin, so it's able to be transcribed, whether there's active chromatin marks nearby, and whether there are regions in your location that may affect the 3D architecture of the genome. And using that, ENCODE is able to predict whether certain regions can act as promoters or enhancers. And so what we see in this duplicated region is that there's duplication of 5 like regions, which would lead us to believe that there will be a substantial increase in transcription. And as Dr. Plan mentioned earlier, some of these basic 3 samples have undergone additional research studies, and this sample is one of those. So we are just beginning to look at RNA-seq data to further investigate whether this variant does play a role in the transcription and potentially implicating this child's cancer. From all of this, we've shown that we've developed a pipeline with the aim to better identify these germline de novo events. And we have identified a few particular de novo heterozygous duplication that we're currently following up on. And we've greatly benefited from the additional research studies involving both tumor and germline sequencing of phase 3, which doesn't stop with the whole genome sequencing. Yeah, so as Dr. Plan mentioned earlier, first, we've been selected for long read sequencing of basic 3. And this is greatly helpful for the identification of structural variants, because in short read whole genome sequencing, there's a problem with poor mapping. They don't map very well to low complexity regions of the genome. And so by performing long read sequencing, we'll be able to identify more structural variants. And so we plan to use a similar methodology to what we use in our short read pipe, but instead we'll only be using one structural variant caller. That's called Sniffles. It was developed here at Baylor College of Medicine in the Human Genome Sequencing Center. We'll then prioritize de novo variants and those that do not occur in a healthy population. So the goals of all of this study is really to compare the 2 results of this structural variant analysis. And it will enable us to identify the limitations and accuracy of our short read structural variant pipeline, as well as structural variant calling in general in trio studies, and as well identify causative structural variants only found via long read sequencing. I want to thank Gabriella Miller Keats First again for this opportunity and this opportunity to talk.
Great. I think that's just questions. Yeah.
Great, thank you so much. Dr. Pong, you already have a question from Dr. Spector. Do you see it, or do you want me to read it out?
Yeah, I don't see it, so that'd be great if you read them out.
Sharon, are you surprised that bi-allelic recessive variants are rare in pediatric patients? ? My impression is that they are usually accompanied by clear phenotypes, so their prevalence had already been established prior to the sequencing era.
Yeah, I mean, Logan's right about that. I think what was not known is that there are hundreds of them, right? So each of them are individually rare. So like Roth, right? Lisa Wong started with me and now is her career, and I think at Texas Children's, we've still, in those 20 years, maybe 2 or 3 additional cases. So we know they're rare, but there are dozens of DNA repair disorders. And the question was, if you sort of add them up, would there be more? I think they don't all have phenotypes. So ataxia telangiectasia, for example, eventually have a phenotype, but children can definitely develop cancer before they ever develop ataxia or telangiectasia. So, and that's definitely true of some of the other DNA repair disorders. So it is true that we knew that each one individually was rare. I was surprised that, for example, that there weren't more undiagnosed Fanconi anemia patients, because that's another one where the prevalence of the birth defects is highly variable. And there are a lot of patients with Fanconi anemia that might not have been detected to have that disorder before they developed cancer. But yes, I mean, the gestalt is that they are rare, but I thought they might have made up at least a few percent. And more of these studies are suggesting it's probably less than 1%. On the other hand, I should say in our study of rhabdomyosarcoma, we were surprised to find that about 1 in 5 of the 500 patients appear to have Costello syndrome. So that's an example where what we thought was really, really rare may in and of itself make up 1% of rhabdomyosarcoma patients. And so it was a little bit more that kind of data I was expecting than what we did find.
Thank you. You have another question from Laura Eagles. What data sets did you use for filtering against control populations for the SV study?
Owen, I'll let you take that if you haven't disappeared.
Yeah, I'm right here. So for the structural variant, we're initially just doing the other unaffected parents for our initial healthy control population. But I'm sure as many of you know, NOMAD recently came out with a large structural variant pipeline on what appears to be non-affected, non-cancer affected patients. So we're also comparing those variants one-on-one to that, but the initial filtering is only against other unaffected patients. One of actually the nice things about the long-read pilot is Genome Sequencing Center here at Baylor does actually have cohorts that undergo the same long-read sequencing that our samples will also undergo. So we'll actually serve as a very nice healthy control in that sense as well.
And for your finding, now that you've been able to look in NOMAD, has that been called?
No, it's not. So it's completely novel.
Okay.
You had another question that, oh, go ahead.
Oh, yeah. So how many patients are you aiming? So we are aiming for roughly the same amount who underwent short read whole genome sequencing. We are limited in part by the amount of DNA that's required for long read sequencing. It's quite a bit more. But from initial estimates, we can get roughly, I would say, hoping, fingers crossed, 60 of the 67 we originally sent for short read whole genome sequencing.
Well, and I should say, because Valerie will probably correct us, we've been approved for the pilot, which is much smaller, and then we are likely to be approved for the longer one, assuming the pilot goes well.
Yeah, that's right. And I will talk a little bit about this overall later in the webinar, but all the numbers are sort of TBD, you know, because of the QC at play. But yeah, you guys are targeting a pretty large sample, so we're excited about it. Any other questions? Hopefully everyone's figured out how to use the Q&A panel on the bottom right. Any other questions for Dr. Plan or Owen? Okay, well, Dr. Plan, we know you have to go, but if we do get questions for you later in the webinar, we'll make sure to follow up with you separately.
Right, and I'll stay on for a few, but then yeah, I have to go do an education thing, so. Thanks again. And I assume you will take back over computer.
That's right. I'm going to turn it over to Adam Resnick as our next presenter. And thank you so much for that really wonderful presentation. You know, I also want to just comment briefly that usually we try to do these in September for Childhood Cancer Awareness Month. So we just barely missed it, but we really enjoyed, you know, getting that really in-depth presentation from you.
Thanks.
Okay, Adam, and I'm unmuting you, Adam, so you should be good to go.
Okay, great. Thanks again for joining this webinar. Hopefully some of you have joined previously. As Valerie mentioned, we've done several webinars previously over the course of the last 3 years of our tenure as a data resource center for the program. Today I'll try to just review where we are and contextualize it a bit, and then turn it over to Vincent Ferretti, who has really been leading some of the portal-based developments, and he'll highlight additional features that we've been developing. To provide you a little bit of context, most of our efforts really center around this overarching methodology that we try to bring to the Kids First data resource infrastructure and practices, really recognizing that we have to cover this end-to-end solution that empowers the transition from local based efforts, oftentimes undertaken by investigators or their consortia, to really providing the platform-based initiatives, this translation of those samples and the data generated from those samples by the sequencing centers into structured information that can then be harnessed by the community for knowledge creation and ultimately patient impact. And part of the reason I wanted to remind those of you who haven't, or if you have joined before, but remind the audience of our methodologies, because you'll hear also later from the Common Fund data ecosystem. And one of the most exciting things that's taking shape within the NIH landscape is really this double funnel paradigm that we've been trying to embrace, where on the front end of the research lifecycle, especially within the Kids First data resource, you see this large diversity of investigators, data sets, disease contexts being intersected and brought together in ways that harmonization provides a narrowing of opportunity for discovery across the platforms. But then at the same time, the platforms are supposed to, if we're successful, expand the use of such data and expand the impact of such data through greater access to the community. But what's also exciting is through interoperability with other platforms. And it's really through this life cycle that we think we can really drive accelerated impact on behalf of children, really in alignment with the mission of the program. And so it's really beginning in that first phase of taking large-scale data generation efforts that are still ongoing. And one of the more exciting things is that the kind of research that Sharon just highlighted is really moving forward. Valerie also highlighted a few publications that are taking shape. But that's really just the tip of the iceberg, because you'll see that there are release studies and many more studies coming on board. And really, now that the release studies are available, that expands that usage not only to the initial investigators, but also to the rest of the community. So these studies really, as Valerie highlighted and Sharon highlighted, really span disease-specific contexts, but also really integrate across both cancer and non-cancer contexts in ways that's both challenging and filled with opportunity. I tried to summarize just high-level numbers across these different organ system and contexts. And what you can see that this creates some of the most diverse biological contexts for disease in pediatrics, from a whole genome sequencing perspective paired with RNA-seq and some of the long-read studies that are coming up, as well as with the clinical and phenotypic data that's being added to them. Taking that data and really structuring it in a way that's useful, not just for the investigators, but also for secondary use, really takes a combination of platforms that is continually evolving. And at each phase of platform development, we go through a number of cycles of community engagement, of alpha and beta testing, to really assess if we're meeting the needs of the research discovery process. And every one of those cycles is also available for your additional input as a community for continued iteration. And you'll see some of this in Vincent's presentation. Genomic data is, while challenging and large, is not nearly as challenging as the clinical and phenotypic diversity. And we've highlighted some of the tooling around the Kids First data resource centers' capacity to handle and empower clinical phenotypic data structuring. And the empowerment is not just at the querying phase for the users, but also on the ingest phase. And I wanted to just briefly highlight this because some of the tooling that spans that life cycle from end to end is not only focused on the portal endpoint, which oftentimes is what we present, but also on the ingest phase. So for example, here you can see many of the studies begin in essentially Excel sheets or some other tabular format that now we are structuring through different toolings that can support both the real-time tracking of those efforts and what hopefully you'll see in the near future are implementation of additional services around the clinical and phenotypic lifecycle of data sets in ways that I think is really dramatically empowered. What you have heard at each time that we've had a webinar is some of this translation of information into knowledge by the diverse investigators. Our initial mandate over the first couple of years was really just to empower access and structuring of the data to ensure that this diverse data set is really fair, findable, accessible, interoperable, and reproducible. But now we're moving into this additional phase of activity that expands the capacity of both investigators as well as secondary users to leverage the platform for additional discovery. Owen highlighted some of the tooling around the analytic workflows in Informatica, but the next phase is to take some of the processing of that data and empower it for querying and hypothesis generation and discovery in the way that Owen and Sharon just described, but more broadly across data sets and across studies. And this is what Vincent will really focus on. He and his team have really been champions of implementing an initial deployment of such tooling as a variant workbench. This is a really significant milestone because it truly empowers the queryability and assessment of very large-scale data sets when it comes to simply the numerical challenge of such a large number of variants across the whole genome context of many of these studies. We're still in the early phases of the deployment of this resource and expect it to really expand rapidly as we engage both the EXO-1 investigators and the rest of the community for further implementation of these toolings. I wanted to, I'll turn the presentation over to Vincent in just a second, but I wanted to again point out this other end of the funnel, really to connect to some of the presentation content that you'll see from the Common Fund Data Ecosystem team, but also contextualize it in a large body of work led by Alison Heath and others from our team, along with the NIH to really empower the interoperability and access to data sets across a number of platforms. And so that funnel of expansion not only pertains to additional investigators and additional studies, but additional platforms. And it's that phase of expansion that is truly exciting because it will open up the doors for Kids First Data Resource users to not only access and utilize data sets within the Kids First environment, but as we have highlighted in the past, it expands to the NCI, NHGRI, NHLBI, and the Common Fund, providing hopefully a really robust architecture for rapid discovery. We oftentimes lead many of these presentations at CHOP, but obviously we couldn't be here without our really amazing partners, one of which I'll turn this over to right now, which is really Vincent from St. Justine. So thanks again, and we look forward to continued engagement in the ongoing development of these platforms and resources and to support the discovery process, not just for the investigators who have done an amazing job at collecting and supporting these large-scale data sets and specimens for their own research, but really empowering secondary users, as Sharon was really doing an excellent job of highlighting, for the use, integration, and analysis of those resources. And please feel free to reach out to any of our support staff via the website anytime. So at this point, I'll turn over the presentation to either David or Vincent.
Yeah, I want to apologize that we're having some technical issues letting Vincent share his screen. So Vincent, I have unmuted you though. Do you want to do a quick sound check? Actually, I don't hear you, Vincent. Oh, I hear something. Oh, I don't think I can hear Vincent. Oh no. I don't think we're going to be able to hear Vincent. Is there someone else I can turn it over to?
Sure. I can try and do a very poor job of presenting on behalf of Vincent. This is Adam.
Okay.
And then Vincent, if you join or are able to call in, I'll be happy to turn it over to ensure that you do justice to the presentation. As Sharon and Owen highlighted, there are a number of different analyses that can be done on the whole genome sequencing data sets, including structural variations and variant identification. We began with really trying to develop resources to support variant identification, and importantly, to support such resources that would really span the entirety of the data set in ways that could still support the appropriate access and management and use of those data sets in accordance with the consents that a patient provided, but at the same time empower that discovery in a common environment. So the objective is really to extract, annotate, and then import this variant data as processed uniformly by the Kids First Data Resource Center into a queryable data set. Now, this is extremely large, big data, and as Vincent highlights here, really requires us to maintain confidentiality and security in alignment with the consent efforts that the investigators themselves initially undertook to bring patient into the protocols. The production version will be released in a coming setting with a phase 1 for Exo-1 investigators teams to inform the usage and development of these resources between now and November, with the goal to then expand wider use in the early months of 2021. So we'll leverage really the Exo-1's investigators' expertise and knowledge base within their own studies to ensure that across these different resources, data sets, the variant workbench can really support the required filtering and annotation and discovery processes that can then be more broadly expanded. Our vision is in the future that this will also be available for investigators, just like Cabataca is, to support the ingest of additional data sets by investigators, harmonizing such data sets and incorporating the investigators' own variants into the context of a broader variant setting in alignment with the appropriate consent and practices and governance. So really bringing these together requires intersecting the variant data sets, and given all the effort we spent in annotating participant clinical data, we want to make sure users can not only query variants, but also intersect with sophisticated variant querying that is mirrored by their activities in the cohort building portal sections that we've demoed in the past. And then we've also integrated additional public variant data sets in order to empower the calculations and understanding of any particular cohort within Kids First. Each user actually has a dedicated instance of a Spark cluster that gets initiated within the Kids First data resource architecture that retains and is persistent across their analyses, as supported by a Zeppelin notebook implementation in the portal. Next slide. What empowers really this rapid queryability is the organization of the data into a relational database that's tabular in nature. We can continuously add additional tables into this infrastructure that supports really rapid querying across that tabular context within the Zeppelin notebook in ways that allows for very rapid iteration of hypotheses and variables in the context of pre-structured notebooks, or for those users who have more coding experience, even highly complex, independent, and bespoke notebook context. Next slide. It is a continuous challenge, but also a requirement that, as Sharon and Owen discussed, empowering our discovery always requires a comparative linkage to other existing large-scale variant data resources. And to accommodate that within the variant workbench, we have been integrating many of the standard variant annotations from NOMAD, OrphanEd, COSMIC on the cancer side, TopMed, and then intersecting those with the clinical and phenotypic nomenclatures that are part of our own data sets and annotated by additional programs like the VEP and DBSNFB. These are the types of underlying resources that we continuously integrate, and as part of the initial development cycle, we'll be looking for additional input as to other resources that should be included as part of the variant workbench to support additional discovery. Bringing all these together allows you to create really complex variant filtration and annotation across not just any single cohort, but across very large data sets within Kids First in ways that can support finding de novo rare missense mutations, those that have high functional impact, those that then intersect within one data set or another, and the relevant frequencies as tabulated across appropriately consented cohorts. Next slide. So currently, we have loaded 11 studies with a little bit more than 11,000 participants. These include almost 300 million unique variants across 53 billion occurrences. Again, these are our goal here is to really provide an initial starting point for the filtration, analysis, and annotation in ways that is dispassionate from the perspective of the Data Resource Center. We wanted to ensure that the appropriate filtration and analyses are really informed by domain experts and investigators. And so really these are the starting points of the outputs of many of our pipelines, but now this environment can support that analysis. So as you can see here, each variant occurs on average 178 participants. Each participant has a very large number of initial called variants that need to be filtered for analysis. And then as you can see, these overlap significantly with other resources like TopMed or NOMAD. Next slide. Key to the way that we and Vincent's team have been developing the portal environments is to try and create a singular environment as a starting point for the research activities. So the Zeppelin data analytic environment is actually integrated into the portal environment and is dedicated again as a safe state to the individual who has logged into the portal. And our goal is to make this environment available to any user who logs in to the portal, the same way that Kabatica and the clinical phenotypic data are available for any user at this time to query and create cohorts, recognizing that oftentimes these activities that then support additional data use requests and additional activities. But in order to perform these activities, you will have to have a Dbcap access permission, in contrast to the somatic data from cancer, which the NIH at this time still supports as a accessible data set in the absence of a Dbcap request. Germline variants obviously provide additional risk, and as such, you will still have to have the appropriate Dbcap approval, but any Dbcap approved study that you have can be performed then within the context of the variant workbench. Next. We're leveraging the Zeppelin notebook environment for a variety of strategic and tactical reasons. This includes our capacity to create really standard variant workbench environments as starting point for investigators who may or may not have significant experience with notebook environments, leveraging the graphing and charting capacity innate within the Zplin notebook environment. And then at the same time, allowing more experienced users to really have a very wide range of choices in programmatic language implementations across the notebook environment. This is a continuous challenge within the Kids First Data Resource Center's efforts to really allow both the high expertise of bioinformatician and data scientists, but at the same time, make really complex data approachable to the biologist and bench scientist like myself. It's in this environment that you could then really choose a variety of different analyses, and then at the same time share these analyses, create and store multiple different versions of notebooks, and create a collaborative environment that leverages both your own data as well as the DRC's hosted data sets. We do have additionally some pre-installed libraries like Kale, Pandas, and others. But again, as part of the initial alpha and beta phases, we'll look to optimize around those resources as they're available. Next. Here, one of the main underpinnings for us was to ensure that the cohort creating capacity of the portal, which we spent a large amount of time vetting and optimizing, can be leveraged to drive this additional discovery for secondary use. So again, we're entering that secondary use phase of multi-study investigator initiated and hypothesis-driven querying. And so as users create cohorts across data sets that they have access to with the cohort building activities, those cohorts can be saved, shared, and analyzed by multiple users, but then can be handed off as a saved participant set to the variant workbench environment. And so this combination of having saved cohorts and saved workflows and environments can be paired in multiple different scenarios, right? So a user can iterate on the cohort creating abilities once they do an analysis to further highlight any feature they detect within a particular cohort, and if vice versa, a user can iterate on the analysis side by pushing a saved participant set into a variety of different workbench workflows. Next. These saved sets essentially are your own personalized cohort that is persistent and allows you to be joined with all the other variant tables for additional genomic analysis. And it's this concept of saved sets as a saved state for a user that we really hope to continuously iterate on and continuously develop a workbench workspace environment for users to return to the portal and analyze. Downstream features that will be implemented that we hope to bring on board include notifications around such saved sets and workflows, such that the dynamic context of ongoing data generation and incorporation into the Kids First DRC could be reported to users across their saved sets and or workflows. So you can imagine as we onboard and intersect additional data sets across this highly variegated data landscape, the cohorts themselves and variables initially selected will change, as well as the significance and analyses that are taking shape within the variant workbench. And our hope is that having such saved sets and states will allow users to iteratively be notified as the data lifecycle continues and additional data comes into the resource environment in ways that is highly flexible and informative to users. Next. I addressed some of this, to be transparent, it's one of the most vexing questions that we oftentimes think about within the Data Resource Center. And that's because, to really drive the kind of research you saw from Sharon and Owen really takes a large amount of expertise at the bioinformatic and data science side. But at the same time, the scientific hypothesis generation by biologists, clinicians, and other stakeholders need to be supported. So for us, the workbench is really for, initial data quality control that are rapidly available for any users to assess their data sets. But then we'll also support via standard workbench notebooks the initial analysis and queryability of data sets or variants. And then ultimately, our hope is to drive the continuous improvement and implementation of much more sophisticated bioinformatic workflows as generated by the community, bioinformaticians and data scientists who are SQL, Python, R, et cetera, enabled. So this will be an ongoing experiment within our efforts to really try and provide these services across both QC, initial queryability, across very large number of variants, as well as much more empowered, sophisticated notebook environments. So our next aim is really to increase the user base so that we can both learn and optimize on the deployments of these environments, and users will be able to both change, modify, implement, store, break, and improve upon this implementation. And as Vincent highlighted in this last point, it's really a trade-off between sort of standard data portals that have static implementation within their portal environment. Within Kids First, for example, we have a PDC Bio instance that really is a great initial queryable environment, and the much more sophisticated, but oftentimes more limited in terms of its user-based environments of programmatic interfaces or command-line environments. So this will be an ongoing effort to really position this workbench across that domain, and features that are reusable and developed with the workbench will also ultimately end up as static and empowered queryable environment within the portal itself. So it provides a great way to pipeline additional implementations for us. Next. So there's, as I've, I think, repeatedly highlighted, is a lot more work to do, including the indexing of variant database with Elasticsearch, building data querying interfaces within the portal itself that leverage the development cycles around the Zeppelin notebook environment, and then also provide more routine, what Vincent calls nomad-like interfaces that return really aggregate high-level views as appropriate for a data set across the Kids First, really growing body of data. And then further integration with the cohort builder that allows for complex queries, not just across phenotypes, but across genotypes within the portal itself. So this will be a continuous development cycle that will improve the user experience and hopefully the discoverability of meaningful results. Next slide. So undoubtedly, I did a poor man's job at presenting on behalf of Vincent. Again, apologies to the St. Justine team for the technical difficulties. Hopefully, I did it some justice, but not sufficiently complete. We'll have plenty of more opportunities to demonstrate the use of these portals, particularly in the context of actual analysis on data sets in coming webinars and deployments. But really kudos to the St. Justine team for doing an amazing job for this deployment. Thanks again to the amazing CHOP team for making it possible. And especially thank you to the investigators who, whether or not they knew it, have volunteered to be initial guinea pigs and users of these resources. And we look forward to working with you and the rest of the community as we try to empower much more accelerated discovery across really amazing cohorts of data. Thanks.
Thank you so much, Adam, and thank you for being willing to do that without notice. And I do want to apologize for everyone. We don't know what the issue is, but we can work with the team to maybe pre-record a variant workbench demo and make that accessible to the community. But with that, our next speaker is Titus Brown from the Common Fund Data Ecosystem. And Titus, you should be able to unmute yourself and share your screen now.
All right, I'm unmuting. Let me see if I can share my screen.
I can hear you. Good.
Yay. That's the first step. And then let's see, where is the share screen on this?
There's like an arrow button at the bottom.
Share my screen, screen 2. How's that?
I can see your screen. You might want to make your slides full screen. That's presenter mode.
How about this?
There you go. Perfect.
Excellent. All right. All right. Hello, everybody. I'm going to tell you a little bit about the Common Fund Data Ecosystem. This is a new NIH initiative that was going public at the end of the month. So, and you can find out, just to start off, you can find out more at nih-cfde.org, and I'll give you some more links and URLs and stuff later on in the talk. So this is just going to be an overview with some points of relevance for the Kids First community. But before I get started on that, I want to say that I'm here representing a much larger group of people. The CFDE is led by, the Common Fund Data Ecosystem is led by Owen White at the University of Maryland, Baltimore, and includes a large number of other people, including Avi Mayan, Susanna Sanson, Carl Kesselman, Ian Foster, and others. I'm the lead of the engagement and training component of the Common Fund Data Ecosystem, and that's a lot of what I think is of relevance to the Kids First community. So the overall mission of the Common Fund Data Ecosystem is to increase data reuse across Common Fund programs. So we want to enhance both the reuse of data within individual programs, as well as reuse and comparison across various programs. There are a number of Common Fund programs that are both currently being funded, but also many that have been sunsetted, and we want to maintain access to those older programs, data sets. We also want to broaden the application of tools across different CF programs. And of course, at the end of the day, what we really want to do is enhance the ability of everybody in the community to ask scientific questions across Common Fund data sets. And of particular interest to me is the idea of increasing the on-ramp for more researchers training. And so this sort of dovetails pretty nicely with the end of Adam's presentation, where he talks about the trade-off between tool complexity and applicability to users. So just to continue with the introduction of why the Common Fund Data Ecosystem, the Common Fund supports a pretty diverse portfolio of projects: 4D-Nucleome, GTEx, Human Microbiome Project, HubMap, which is a single-cell RNA-seq program, focus program, Kids First, Links, which looks at drug targets, metabolomics, MotorPack, which is looking at the impact of physical activity on many different molecules, and also Spark, which is looking at the peripheral nervous system. And these various programs produce a wide variety of different types of data, some of which, in particular transcriptomics, as you can see here on this slide, are shared across many of the programs. And so the idea of the Common Fund Data Ecosystem is that we want to start connecting programs by these data types. So here's an example upsetter plot showing the number of, for example, the number of samples from the number of tissues for which we have samples per program by CF program. And so you can see, for example, here that there are 11 tissues that are at the Human Microbiome Project and are shared by at least one other program that has data from that same tissue. So we can't yet do this on the fly across all of the programs, but we're working to connect the data sets, connect the programs and the data sets in order to make this kind of information dynamically discoverable. So the idea is you'd be able to find data sets of interest in, for example, Kids First, and then do a sort of pivot search or a pivot query to look across other Common Fund programs to find similar or relevant data sets, such as across GTEx. So here, for example, you can see a list of the 11 shared tissues in this case. So, The vehicle, gets a little bit technical, but the vehicle by which we're working to connect across programs is something called the crosscut metadata model. And I think the real point here is that the CFDE is not interested in storing all of this data. We're not a data warehouse. We're not replicating data across from across the various programs into our own space. And we're actually largely hoping to function to redirect users to the various Common Fund programs as their primary resource. So we're hoping to serve the data coordinating centers, the data resource centers, by connecting them to other Common Fund programs and directing users to the individual programs. So you can see down below here, there are a variety of locations that have RNA-seq invariant files, and we want to connect those via the CFDE catalog across the various programs. Here you can see our alpha or beta website for the Common Fund Data Ecosystem web portal, where we sort of give summaries of the various kinds of data sets that are available, publications, news, that kind of thing. This website is open for business. It's at nhcfde.org, and I'll point you at two specific parts of this in following slides. One of the things that we're producing across the Common Fund are dashboards of the various kinds of data sets, anatomy, assay, RNA-seq data sets, and so on. Our goal is to have these be up to date as the various programs produce more data, and eventually we hope to connect this with more programs beyond the Common Fund even, because we're developing standards and so on that let us share this kind of information, these kinds of catalogs across a much broader range of programs. So there's a public beta that will be open at the end of October. We have a CFDE portal for cross data set search. The idea here is that you can do things like keyword searches for various kinds of data sets, various types of files, and so on and so forth. At the beginning, this won't be as powerful, for example, as the Kids First DRC portal search in terms of disease and diagnosis and detail and molecule types. But we're going to iterate on this and slowly build it out so that you can do single location searches across a really wide variety of data in order to find data sets of interest. The overall mission, again, is to increase data reuse, and a really important part of that, and something that is the reason that I signed on, is that we're really interested in increasing the on-ramp for more researchers using training as a vehicle. So we view user training as a critical tool. We're going to be providing user training using DCC-specific resources. So, for example, I'll show you in a slide or two, we have some initial training set up for the Kids First DRC portal in order to connect it, to find various things and connect with various resources. We're hoping to develop training for resource discovery across Common Fund data coordinating centers. We're also very interested in providing training to analyze data sets in the cloud using, for example, the Kvatica platform. We are working on data type specific training. Hang on a second. Working on data type specific training, for example, whole genome shotgun sequencing data analysis for clinicians. We have several MDs on our team who are contributing in that area. And our training modalities at the moment largely are websites, videos, and teleconference training. Eventually, we hope to do in-person training again. Our initial training plans were actually focused on running in-person workshops at various locations, but with the pandemic, we have, of course, suspended those plans. So in terms of Kids First, most specifically, we're planning tutorials and training on a variety of different things, logging into and using the Kids First DRC portal to find data sets and making full use of the query system that the Kids First folk have put together, running various data analyses in the Kvatica cloud platform and or using various specific apps for whole genome shotgun sequencing or RNA sequencing in particular, data type specific training, WGS for clinicians. The modalities, again, right, include all of these different approaches. So what I think is probably going to be the most browsed resource will be our static training websites where we do we have walkthroughs and screenshots, and we're putting those together again for Kids First. So one of the things that we are, one of the other things we're hoping to do is specifically support Exo-1 investigators in the future in a more intensive way. We're still working up to that. Right now, we're focused on getting a handle on all of the different things that we could provide training on. But if you're a current or future XO1 investigator, please do reach out to us, and we'd love to talk to you. With respect to the amazing resources that Owen shared with us earlier, I added this little slide here. We're also happy to talk to people who are developing tools. We have a team that can help you test, document, and develop training materials for new tools. And that can be very useful in terms of relieving the burden of doing all of these different things while you're also developing a novel tool that you're also applying to your own research. And we're happy to help you generalize the tool for others to use. A sort of more nascent program that I think just the funding has just been approved for is that various of the Common Fund programs are setting up to work with each other. So in particular, GTEx and Kids First are hoping to support a more rich and robust collection of cross-program analyses. I believe Kids First has also reached out to HubMap and Links in order to integrate some of their resources more directly into the Kids First portal and downstream data analysis. And we're here to help support them in doing that. So hopefully there will be a much expanded set of functionality coming through in the next 2 to 3 years. Last but not least, I just want to point you at the public training website. We're constantly expanding this. I think our next release is mid-October. The training website is up and available for you to go look at. It's linked from the main NIH CFDE page. Here, the goal of this website is to broadcast modules for learning how to use various CFDE resources. You can see we have a special Kids First tab right here in the middle. And that tab contains, on the right, various Kids First lessons. Right now, the main one is registering for the portal, connecting accounts, and accessing the data. But we are going to be producing Cavatica tutorials in the next couple of months. We also have general bioinformatics tutorials over on the left, and we're constantly expanding those as well. So we welcome feedback. In particular, if you go to the NIHCFDE.org webpage, on the upper right, there's an About link with a contact page. And in the contact page, you can drop us fill in, there's a free text box where you can tell us what kind of training you're interested in, what kind of data set combinations you're interested in, or if you have questions or comments or requests, we'd love to learn what we can help you do. So I think, I guess that was a rather rapid volley through the slides. I'd be happy to take any questions if we have time, or I'll let Valerie let me know what I should do.
We can pause for a moment and see if any questions come in through the Q&A.
Sure. While we're pausing, I'll just say, please feel free to reach out to me directly. I'm CT Brown at ucdavis.edu. I'm pretty easy to find. There's only so many Titus Browns on the internet.
Okay. You know, Titus, I'm not seeing any come through, so I think I'm going to take the presenter back. But, you know, stand by in case something pops up. And if something comes in while I'm talking, you can actually even respond and type a response to the question.
Sounds good. I'll go on mute now.
Thank you. Okay, let's do our final session. So in case you all forgot where we were on the agenda, thank you, Titus, for that excellent overview of the Common Fund data ecosystem. And I think we're very excited to work with you. I think that training for the research community is a really critical part of what we've been trying to do as we launch all these beautiful tools that the data resource is building. And so we look forward to getting your help with that. Now it's my turn again to provide some more updates about the program. You heard a little bit about this already, and in the spring I told you that we were planning to launch a long-read sequencing pilot this year. This is working with our Kids First sequencing centers, the Broad Institute and the Hudson Alpha Institute for Biotechnology in collaboration with St. Jude. The goal of this pilot is to leverage long-read sequencing technologies such as those offered by PacBio and Oxford Nanopore, and the expertise of the sequencing centers to further inform findings from Kids First projects. And I'm happy to announce that we will be providing long-read sequencing to these Kids First projects to identify structural variants, which you heard a bit about, but they are large genetic aberrations such as deletions, duplications, inversions, or translocations. And these are typically difficult to detect with standard short read whole genome sequencing, which is what the program has primarily focused on so far. But these structural variants may actually be causing these conditions in some cases, and the long-read sequencing could potentially inform the discovery of new therapeutic targets. So briefly, the non-syndromic craniosynostosis project, congenital diaphragmatic hernia, structural heart defects and related defects, the bladder atrophy epistates complex project, the acute myeloid leukemia project, novel cancer susceptibility, which was Dr. Plan's project that she spoke about today, and the project that focuses on Ollier disease and Maffucci syndrome. You'll note that the short read data sets for some of these projects are already released to the public, so you could, access them now and begin your analyses, and the rest of them are coming. We used a variety of criteria to select these projects and to allocate resources for a wide range of numbers. So some of these projects, we're really just starting with a few trios to see, what could be found in really critical cases. But in some of these projects, we're actually aiming to do up to 200 samples worth of long-read sequencing. And that is the goal for Dr. Plan's project as well. But all of that is very contingent on whether the DNA is of high enough quality and whether the DNA fragments are long enough to actually detect these variants. So we will be publishing a web page that summarizes the individual details of each of these projects. We weren't able to get it up before today, but stay tuned. It will appear on the Kids First Common Fund website. Okay, strategic planning and phase 2 initiatives. So as you may remember from previous webinars, we have solicited and compiled feedback from the community, external program consultants, NIH leadership and staff to inform our plans for the last few years of Kids First. This feedback coalesced into 7 consensus recommendation themes, and even prior to developing our phase 2 plan, it really helped guide us in thinking about our priorities and our collaborations over the years. But of course, it did feed very directly into the development of the initiatives that we proposed for phase 2. James Coulomb, our working group coordinator, gave a more complete presentation of this process at the recent NIH Council of Councils meeting in September, and the recording and the slides are actually available to all of you to view. I do want to also say we'll give you all of these slides from today, so you have all of the links that are embedded in these slides. But to summarize James's presentation, the thesis was basically that the data resource has already proven to be of high value to the pediatric research community. And the proposal for phase 2 aims to further enhance that value and impact of the overall program, as well as the data resource. So with the $12.6 million annual appropriation that we get in Kids First, for the last 3 years of the program, we proposed 3 broad initiatives. The first one is to continue generation of data from childhood cancer and structural birth defects cohorts, but include the addition of epigenomic and proteomic assays, to continue improving and developing the data resource, and to start looking towards how to sustain this resource and its impact beyond the end of the 10 Common Fund years, and to engage experts in activities that increase the value of Kids First data sets. One prominent example includes activities such as the integration, curation, and harmonization of rich clinical and phenotypic data sets that are associated with the whole genome sequence data that we've been focusing on generating thus far. So these concepts together aim to build on the infrastructure, the data sets, and the expertise of the rapidly expanding Kids First community to strengthen data sharing and collaborative discovery, which is of course in line with the program's vision, and these concepts were approved by the Council of Councils in September. And of course, in parallel to these new initiatives, we will continue our existing collaborations, and we will look for new collaborations as they pertain to our goals. We have quite a few collaborations. I've spoken about many of these in the past. They not only amplify the overall impact of the program, but they help us address gaps where we're not quite able to. So today, I'm just going to give you little updates on our, a couple of these, starting with the interoperability efforts. In the spring, I told you that the Kids First Data Resource Center, NHGRI Anvil, NHLBI Biodata Catalyst, and the NCI Cancer Research Data Commons, as Adam had already alluded to, are working together on technical solutions to enable researchers to combine and cross-analyze data sets that reside in these different NIH genomic data sharing platforms. The NIH Cloud-Based Platforms, or NCPI effort, is being driven by use cases that we hear from the research community. And for Kids First, we are focused on federating access to a variety of childhood cancer, structural birth defects, and other pediatric data sets. Many of you may be aware that Anvil, for instance, is hosting the Centers for Mendelian Genomics data, overlaps with quite a few birth defects that we're studying. Congenital heart disease data sets accessible in NHLBI, for example, and of course, pediatric cancer data sets that may be part of NCI efforts. We're also in the early stages of exploring interoperating with additional data sets even beyond these 3 other platforms. The NCPI group recently worked with the Office of Data Science Strategy to establish a web page where you can learn more about this effort, including the issues we're tackling and how we're going about it. It's pretty technical, but if you want to learn more, I encourage you to check out this link and read about what we're up to. Okay, and I also want to update you on our phenotyping and cross-species collaborations. To guide us in our phenotyping efforts, the Monarch Initiative and the Data Resource Center have been working together to host a series of webinars called the Innovation Across the Phenotypic Translational Divide. And the goals of these webinars are to improve phenotype data collection and harmonization of Kids First data sets and brainstorm technical solutions for facilitating genotype-phenotype analyses. We've convened Kids First investigators and other researchers twice so far to hear about the variety of phenotype data represented in Kids First and to bring light to some of the opportunities we can work on to enable cross-disease analysis. The hyperlinks in Part 1 and Part 2 are summaries, blog posts that summarize what each of those webinars tackles, basically. And you can learn more about the webinar series at this link as well. And we plan to work with the DRC, the Monarch team, and the COMP2 program to continue this momentum with a Part 3 of this webinar series. This one will focus on opportunities for fostering cross-species analysis, for example, how to use model organisms and associated databases and tools to validate the function of variants identified in Kids First data sets. And as Titus mentioned as well, there are opportunities for the Common Fund data ecosystem to sort of facilitate some of this as well. So we're still developing the agenda, but we have saved December 9th as the date, so feel free to mark that on your calendar. And as we solidify the details, we will announce it through our listserv. Okay, and with that, I'd like to kick off the Q&A period. So I still need to open up the Q&A so I can monitor it. But allow me to start by answering some, or David and I together will answer some questions about that we received prior to the webinar. So let's start with what support is available for learning how to use the Kids First platform. And David, are you able to unmute yourself?
Yeah, I'm here. Can you hear me?
Yes.
Great. Yeah, so one question that we get, especially after these webinars, is what types of support opportunities and tools are available to learn how to use and access these platforms. And so Valerie, you can hit the next slide for me. Thanks. And so I would say one of the best places to get started is on our support pages. So we recently rebuilt our support pages from scratch, and now we have a new set of documents, and they're located right on our website, kidsfirstdrc.org. And then right at the top of the menu there, you see the big blue support button. And in that drop down, that will take you to our support pages. And so on the next slide, we've got a image of one of those pages, which might be a great place to get started. So this is our Studies and Access page. And so here, especially if you're interested in learning about what are the latest data sets that are released, how do I get access to them, what are the Dbcap study pages for these different studies, they're all listed here on the Studies and Access page. So this was a great page to keep in your browser and keep visiting as we continue to update our studies. And so like I believe Valerie said, we've got 14. We're looking to continue to add a couple more hopefully before the end of the year. Okay, so if you look through the study pages and the support pages and you still need a little bit of help, then Valerie, if you go to the next slide, then the next place that I would hope that you point to is our Kids First support inbox. And so you can email us any questions at any time at support@kidsfirstdrc.org. So we've got a couple different people who monitor that inbox here at the DRC, and we review it several times a week, and we look through the different emails, and if we can't answer them ourselves, then we try to point them to different folks in the center to be able to continue to support your research and answer your questions. And so the support inbox is great because it allows anybody to email in anytime, but sometimes I know, especially in this age of social distancing, having some human interaction is always good too. And so another great place to learn about the Kids First platforms is this new series that we're running, which we're calling Kids First Office Hours. So maybe perhaps like an office hours when you were in college. What the Kids First Office Hours is, it's a monthly support session hosted by me and other members of the DRC. And so when are these sessions? Office hours are held at 3 p.m. Every month on the second Tuesday of each month. And who can show up? Anybody can show up, and that's what's the great thing about it. And so we've had questions from, very basic, how do I run a query, all the way to much more advanced, how do I need help building and developing my bioinformatic pipeline. So it's intended that anybody can show up, all are welcome, and so please, we encourage you to support. And of course, these supporting efforts are going to be, as Titus introduced about the Common Fund data ecosystem, all of our support will be tied in that and connected very well. So our next session, so when is our next session? So our next session is October 13th, that's 1 week from today. In fact, it's 1 week from today, right about now at 3:40 in the afternoon. And so how do I attend? So you can send me an email to my email address right there, higginsd@email.chop.edu, and I'll be able to provide you with login information. If you email the support inbox, we'll be able to get you set up there as well. But ultimately, we have a lot of resources to try to be able to help folks, and we hope to be able to continue to do that moving forward.
Okay, back to me. So these are frequently asked questions. So apologies if you've heard the answers before, but we still get them. So I'm going to go ahead and start with what funding opportunities are available. So currently, the funding opportunities that are still running is the Kids First Exo-1 sequencing opportunity. We had, approval to continue this into fiscal year 21, which is now, as of this week, and but note that the FY21 announcement is not published yet, but we will be reissuing the announcement that was PAR 19390. So we encourage any teams that have collected DNA samples from cohorts of children affected by childhood cancers and/or structural birth defects to consider applying for this, to review the opportunity for what would be required. In general, we're looking for DNA from the patient and their parents. We also have had proposals where there are multiple affected family members per family, so it could be a larger family if you have a scientific justification for that. And also whole genome sequencing the DNA and RNA from the tumors. So that also includes some exome sequencing and RNA-seq for affected tissue if it's a structural birth defect cohort, and of course the tumor if it's a cancer cohort. All of the materials from last year's announcement are still available on the FAQ page, and we do strongly encourage you to review that. We will make only, we're expected to make only minor changes to the announcement, but the general expectations are the same. I'm also going to take this opportunity to answer a question I got in the Q&A, which is, you mentioned high-value data sets and what are the metrics you are using to determine the value of a data set. So these applications are peer-reviewed, but we also have a series of programmatic criteria that we describe very clearly in the funding announcement and the pre-application materials at this hyperlink. But I do want to point out that some of our greatest considerations are the ability to broadly share the data, and then of course the overall value of the data set in the context of all the other childhood cancer and structural birth defects data sets that are already represented in the data resource. I should take the opportunity to just say, make sure you're on the Kids First listserv, because that's the venue we use to announce the next iteration of the Exo-1 when it is open. We have developed an FAQ for other funding opportunities that you may be interested in. We have a section about analysis grants, and again, we have a R03 that is specifically designed for analyses of kids first data sets or development of tools and workflows to support those types of analyses. But other NIH announcements like the Parent R01 or Parent R03 are also available if that's a good fit for your project. I should also point out that the Kids First Analysis R03 here, it has to involve the analysis of a Kids First data set, but there could be co-analysis with another data set as well. We also have a section about ways or mechanisms to support functional validation, as we talked about, using model systems or model organisms as a next step in understanding the variants that are uncovered in these data sets is really important. So you can take a look at these announcements and again, make sure you're on the listserv. The second most frequently asked question we get is how do I access data? I think you heard a good amount about this today, but just to summarize again, anyone can log into the portal to search across the data sets that are available. But in order to access the individual level genomic data, you will need to find the data sets either in the portal or you can even look at the Dbcap listings on our Kids First X01 page. You will need to submit a Dbcap data access request, which will go to the Kids First Data Access Committee. And once you have access, the resources David described will tell you how to take the data from the portal and push it into a Cavatica workspace. If you've never submitted a Dbcap data access request, you can watch this video, and then this PDF is also an overview summary of what is required. How do I get involved is our third most frequently asked question. I think you've seen some of these links thrown at you today, but of course the support email goes to the Kids First CRC here. You can email us at the program at kidsfirst@od.nih.gov. There is both a Common Fund Kids First website, which is the NIH website about the overall program, and a Kids First DRC website, which talks about the activities of the Data Resource Center. And of course, the portal where you can search the data. Okay, with that, we are already getting Q&As, or we're getting Qs. We'll give you the As. Let me see. Titus, we have a question for you, hoping you're still on. How was the CFDE funded?
Hi, Valerie. That's the entire. Sorry, maybe I missed.
That's one question for you. How was the CFDE funded?
The CFDE is funded by the NIH Common Fund through an other transactions agreement.
Okay, thank you. Does Kids First provide any external facing APIs? Let me upgrade Allison Heath. Maybe David can get started on answering this. I'll upgrade Adam back and Allison.
Sure. I know that as far as I may have to actually turn this over to Adam and Allison. But as far as our platform, so the Kids First portal is synced up with Cavatica, which is our cloud-based analysis platform. And in order to be able to do your analysis, you'd move your files from the portal over into the Cavatica environment. So Cavatica is built by a company called 7 Bridges. And the 7 Bridges has developed their own API, which allows users to be able to operate working remotely or working from the command line to be able to move files, copy files, run tasks, and run analysis in the cloud on the Cavatica platform. So yes,
Yeah,
so
thanks, David.
Yeah, and I'll turn it over to Allison. Yeah.
Yeah, it's a good question. So we have a couple APIs that follow some of the GA4GH standards that are implemented by Gen 3. So these are the DIRS endpoints, as well as pretty standard authentication and authorization. Building the portal, there's a pretty high performance GraphQL API that I think at this time I wouldn't recommend unless you're a very advanced user. Over the next quarter, we're rolling out a new set of FHIR APIs, which will be the API standard for our clinical data moving forward, hopefully early next year. And then as David said, for the workflows and bioinformatics work, Cavatica has pretty nice APIs with pretty thorough API documentation.
Okay, Okay, hopefully that helped. For a clinician interested in kids first data sets, how would they go about getting access? So I can start off by trying to answer this question. Again, the portal is open to anyone. If you have Google or an ORCID login, you should be able to register with that account, and you will have to agree to a click-through agreement to not try to identify or misuse the data. But once you do that, you'll be able to search across all of the clinical and phenotypic data that's available there. But in order to access any genomic data, you would need an ERA Commons account and to apply through Dbcap. I'm not seeing any other questions come through, but I will wait a little bit longer. Is there a way to get metadata of Kids First Data portal? To get the metadata of the portal. Allison, do you want to answer that one?
I think this is probably towards your previous question about the APIs. So all that metadata, if you mean like the clinical data and the biospecimen information, that's where we're really moving to more of the standard. Currently, probably the best way to get that information is if you go to the portal and you do your different cohort selection and querying. You can go and you'll see a way to download the clinical information, the biospecimen information, and have a manifest of all of that right now. But that's what we're definitely pushing hard towards, making that more programmatic with the APIs.
Okay, I got thanks. I also got a question. Do you know what the status of Anvil is? Could you clarify your question? Is that about when Anvil will be live? And I actually don't know if I'm qualified to answer this question, no matter what it actually is. But in terms of interoperability, maybe Allison, could you comment on the status of our interoperability with Anvil?
Yeah, so I think we've been working hard with that team in terms of making the genomic data available via some of the different standards. I mean, as far as I know, Anvil is live and you can log in and they have some of their initial data sets loaded. But as Valerie said, I don't know if we're the best people to be up to date with that, but happy to point you to the right folks who could.
Okay, thank you. Yeah, I think and from my perspective, the interoperability efforts are very technical, way over my head, but the groups are working really hard on this, and I'm glad we launched that website to kind of let you guys know what the teams are up to. And this fall, I think we'll be digging a little bit more into some of that progress. So be on the lookout for progress on our interoperability with the other NIH platforms. Okay, I actually don't see any other questions coming in. Oh no, never mind. We got that question. Okay, and if that's all, then we will close this webinar. Thank you all so much. It felt like we had done the spring webinar yesterday, so thank you for bearing with us through some of our technical issues today. And if you're interested in learning more about anything you heard, feel free. I'll put that slide up again about the contacts. Here you go. Feel free to reach out to us. And thank you again to Dr. Plon and Dr. Resnick for and Dr. Brown, Titus Brown. Thank you all for your presentations today, and we look forward to continuing to work with all of you.
Thanks, Valerie.